Greetings. In this episode, we will be discussing the building blocks of proteins, amino acids. This figure shows a line rendering of the general structure of amino acids. Amino acids can be described as monomers, meaning they're a single unit of a larger structure made up of many of these monomers, also known as a polymer. Monomers are analogous to links in a chain or Legos being stacked together to form a large tower. Now let's look at the structure in a little bit more detail. I prefer the ball and stick 3D rendering, so sorry to those who prefer the previous one. Here you'll see a central carbon atom, or the alpha carbon, linked to 4 other chemical groups, 2 being the amino acid and the carboxyl group. And if it wasn't clear, the amino group and the carboxyl group, also known as deprotonated carboxylic acid, are why these molecules are called amino acids. I would also like to add that the alpha carbon is connected to 4 different functional groups. And though it was a dark time that most of you probably blocked out, back in organic chemistry you should have learned that this makes the alpha carbon chiral, meaning it must exist in one of 2 mirror image forms, L or D, but only L amino acids make up proteins. Don't worry, I'm all done talking about stereochemistry for now. Now let's move on to the business end of the amino acid, the functional group or side chain. There are 20 different side chains that delineate the 20 different amino acids. It is these side chains with their different properties that allow for extreme diversity among the form and function of proteins. This amino acid chart contains all 20 amino acids, and was the only one I could find that I didn't hate basically in its entirety. You will note that the side chains are broken up into several categories based on their chemical properties. There are nonpolar, polar, positively charged amino acids, sometimes known as basic, and negatively charged amino acids, sometimes known as acidic. The charged amino acids are also considered polar, just an FYI. Beneath each structure is the amino acid's name, 3-letter, and single-letter abbreviations. Sadly, there's no shortcuts. You're just going to have to memorize the structures and names of these amino acids. Hopefully, as you learn more about each one and their roles within the protein and biology in general, they will become more ingrained in your memory and easier to memorize. So let's go through and highlight some of what I think are the important properties of a few of these amino acids. First up is glycine. Glycine is the smallest of the amino acids with 2 hydrogens bonded to the alpha carbon. This makes glycine, air quotes, flexible, with little steric hindrance when incorporated into a protein chain. So that little area of the protein is going to be a little more flexible than with other amino acids. Also, glycine is the only amino acid with an achiral alpha carbon, meaning because of those 2 hydrogens that are bonded are not different, so that it is not asymmetrical. Next up is valine, leucine, isoleucine, and methionine. They all have large nonpolar chains, making them aliphatic side chains. These will contribute to protein folding via the hydrophobic effect, meaning these side chains will have to be tucked away inside the protein away from water for energetic reasons. This is also worth noting that isoleucine has a second chiral carbon. Why don't you go ahead and see if you can find it? I think it'd be a good exercise. Additionally, methionine is one of 2 amino acids that contains sulfur. The other is cysteine. This residue is important because when oxidized in the presence of another cysteine, the 2 amino acids form a disulfide bond, cross-linking the peptide chain together. Tryptophan, phenylalanine, and tyrosine are the next 3 amino acids up. They all contain aromatic rings. These aromatic amino acids are the ones primarily responsible for protein's absorption at the 280 nanometer wavelength. This may seem irrelevant, but this allows for a spectrophotometric determination of protein concentration. Oof, say that fast. Basically what it boils down to is this gives us in the lab an easy way to determine protein concentration using a fast and simple assay. Tyrosine is going to get a little extra love because it's also included in this group along with serine and threonine. These 3 amino acids contain hydroxyl groups, making them hydrophilic, aka water-loving. The biological relevance of the hydroxyl group is that they are targets for a group of proteins known as kinases. Now this group of proteins is very ubiquitous and crucial in cell signaling, making these amino acids and their residues very important within a protein that might be important for cell signaling. Now these cell signaling topics I'm talking about could be cancer related, could be developmentally related. There's a lot of diseases and processes where these are extremely important, and like I said, they're extremely diverse. So keep an eye out for these residues. They're going to pop up again later in your biology studies. I specifically didn't talk about histidine with the other aromatic amino acids because of its biological relevance. The imidazole group it contains has a pKa of around 6, meaning it can be protonated or deprotonated in physiological pHs. This gives histidine a pivotal role in enzymatic activities, meaning it can often be found in the protein's active site. So if this protein is an enzyme, histidine is going to be imperative for it to function, more likely than not if it's within the active site of the protein. Lastly, we have Mr. Derpy amino acid, proline. This amino acid's R group curves around and bonds to the amine group in the peptide chain. Structurally, when proline is incorporated into a peptide chain, it causes the chain to kink with its weird geometry. Thus, proline has downstream effects on the shape and fold of a protein due to this weird geometry. But let's save protein structure discussion for another video. I hope that these little factoids about the amino acids were helpful, and hope you learned something. Thanks for watching.
I hope you enjoyed my lecture. If you wouldn't be caused too much trouble, would you please subscribe? And I will teach you some more.